Identify this might be tough. Identify the receptor based on location.
The receptor.
Oh, this one's kind of tough. So based on location, you had exteroreceptor, interoreceptor, and proprioceptor. So do you think this is detecting things on the outside of our body, the inside of our body, or our body position?
Exteroreceptor.
This is an exteroreceptor, just to put things in, because light is on the outside of our body. And so then we look, identify this specific region.
I'm sorry?
Identify the specific region.
That's not a region.
Yeah, there was different regions of it.
Oh, okay.
Are we talking about like units?
No. So here's your cell body. This is what we call the outer segment. So when we're looking at your notes, and you always want to correlate lecture and lab, they're very correlative. This is the outer segment where you would see the rod shape or the cone shaped portion. Well, if we look at this rod shaped portion, this outer segment is where we have those discs with rhodopsin. So this is where we're going to start our visual stimulus. The light is going to cause isomerization of that rhodopsin from cis to trans. That's going to affect this photoreceptor. Oh, that's right. It was going to hyperpolarize the photoreceptor so that glutamate was not released. So now on your exam, not today, but now after you assimilate lecture knowledge, on the exam I would say identify the specific cell, photoreceptor. Identify the neurotransmitter released from this cell, and of course that neurotransmitter is? Glutamate. That neurotransmitter is released in light or dark?
It's released in dark.
It's released in dark. Nice job. Darkness, because that would inhibit these cells. Identify these cells. Those are your bipolar cells. Identify the structural class of neuron. Structural class of neuron. Bipolar neuron. That's what we looked at previously. Identify the specific cell.
Ganglion cell.
Ganglion cell. These axons form what structure?
Cranial nerve number 2.
Cranial nerve number 2, the optic nerve. So identify the entire tunic or layer indicated here. That would be the nervous tunic. Identify the specific space found here. Posterior cavity, and normally I would ask, identify the substance as well. That would be our vitreous humor. What were two functions of the vitreous humor?
To maintain shape of the retina.
Maintain shape of the eyeball.
Protect inside parts of the eyeball.
And holds the retina in place. It doesn't nourish, it doesn't do anything else, but it's going to hold that retina in place. So on your sheep eyeball dissection, when you had that vitreous humor fall out, that's when some people saw the retina stay on the vitreous humor. Some people just saw it detach away from the rest of the eyeball. Still part of the retina is this specific layer known as the
Pigmented epithelium.
Pigmented epithelium. If you want to call it the retinal pigmented epithelium, that's fine. Just pigmented epithelium is fine as well. Ooh, identify this general layer or tunic.
Vascular tunic.
Vascular tunic. Identify the specific structure.
Choroid.
Choroid, two functions. Of course, it absorbs light and nourishes the retina. Ooh, identify this layer or tunic.
Sclera.
Fibrous tunic, specific structure.
Sclera.
Sclera, and again, two functions. Maintains the shape of the eyeball and protects the inner parts. So if we have a pretty good idea